Welcome to Celluline Science for Everyone. My name is Roger. I'm a scientist living in Montreal, and this is my channel where we talk every day about science that may interest you. Today, we're going to talk about the afterlife, but not about the afterlife that you are thinking about. We're going to talk about what happens biologically to our bodies when we die. Humans and animals in general are able to perform many functions. We are able to communicate, we are able to move, we are able to do a lot of things. So all of the functions in our body take place thanks to the expression of the information in our genes. This process of reading the information in our genes is called gene expression. The gene expression changes throughout our life. When we are in our mother's womb, the expression is different than when we are born, and then when we are a child, the gene expression is different than when we are adults. But what happens with the gene expression when we die? Does it suddenly stop? It stops progressively? Or there are genes that are specifically expressed, that are specifically read when we die? In other words, is there information that needs to be expressed when we die? Let's try to answer this question. There is a scientific article in 2016 in which they analyze how many genes, how much information is expressed, is read after the animal dies. They explore this on mice and on a fish called zebrafish. On the next graph, they try to represent their results. On the x-axis or the horizontal axis, they depict the time after the animal death. In the y-axis, they depicted how many genes, how much information is expressed more after the death of the animal. As you see, in both mice and zebrafish, there is more information read after the animal dies. This gets its maximal values 1 day after the death of the animal, and then it progressively decays. So we can answer the first question. After the death of an animal, the information doesn't stop to be read instantly. In fact, there are genes that get more expressed. There is information that is more read after the death of the animal and then progressively decays. Sidebar. The gene expression that takes place after the death of an organism is called thanatotranscriptome. And how do we do the experiments in humans? Because of course we cannot experiment like we do with model organisms. Well, it has been shown that the tissue that is extracted surgically from a patient behaves similar to the tissue of a cadaver for the first 24-48 hours. So, for example, scientists in 2021, what they have shown is that neurons extracted from the brain of patients with psychiatric problems show a specific pattern of expression of genes. So, there is some information that is specifically expressed when these cells have been extracted from the human and the cells are dying. In conclusion, also in humans, we have a pattern expression that is specific of our death. And what does this mean? Well, we don't really know what this means. We have talked about 10 to 25% of the cells that form our body, the human cells. But what about the rest? 75 to 90% of the cells of our body, in fact, are microbial. What happened with these cells? Remember that these cells constitute the microbiome. We live in a kind of symbiosis with the microbiome. For example, the microbiome of the gastrointestinal tract produces molecules that we cannot obtain otherwise. However, when we die, part of this microbiome loses its nutrients. So these microorganisms will slowly die. In addition, when our body dies, it will stop to release some secretions, cells will lyse, and then they will release toxic compounds, so the microorganisms will change. The microorganisms that grow in our body will change in order to decompose our body. And these changes will take years. And this is really important for the forensic science. Death is a transition, it's not a sharp stop. Our individual cells, despite our whole body have died, they read some genes, some information on our DNA after we die. Also, our microbiome, it is going to slowly change in order to allow the putrefaction of our flesh and the recovery of the nutrients that will be taken by other organisms.